is in that water. And that conductivity is coming from damage to the cell membrane. So when the plants are freezing, it can pop the cell membrane. The electrolytes that are inside the cytosol leak out into your DI water, thus increasing its conductivity for electricity. One of the things that was really interesting when we first started this project, so here's our ion leakage. It's done by percent, so how much leaked when I first measured it, and then you freeze them really, really hard so they're all broken. So you can compare it to how many cells total breakage would look like. And then several different temperatures. Your dashed and open symbols are those that were held at 22 degrees prior to this assay, whereas your dark and closed symbols are those that were exposed to 4 degrees for 2 days. And the leakage percentage is a lot less for these ones that have acclimated, which was a really interesting observation considering this plant has been cold intolerant classified. It is responding, right? Cold acclimation is occurring. It's just more moderate than Arabidopsis. For example, down here we've got -1 to -3. With Arabidopsis, that would be -6 to -9 is where you would see similar leakage. So it is responding just in a more moderate way. We wanted to know, okay, cool, it's responding. Does this response increase over time? And remember, this is electrolyte leakage, so you want less leakage, so like golf, low score wins. You don't want it to be really high leakage. And this is the number of days that they spent at 4 degrees prior to being put in the assay. And 0, right, where there was no cold acclimation, you have like 95% leakage. They're really, really stressed plants. Then 1 day drops, 3 day drops, 5 day, 7 day, 10 day. So they are capable of cold acclimation. It takes about 5 days for 50% of the cells to be okay versus 50% of the cells to be damaged. So that was a nice little benchmark for us. Okay, it takes 5 days, 7 days is also good, so that we could set up our next experiments to use those timings. Soybean are classified based on their maturity group, and while this was based on photoperiod, right, how much light this area gets, one of the things you might notice is this is also based on north-south. So 000s, they're up in Canada, right, and then you have all the way down to Florida, soybeans can be grown. So this also led to the idea of perhaps not only are there different cold tolerances, perhaps it was this selection that also helped with that. So would these northern varieties be more cold tolerant than southern varieties? And would this hold true and hopefully be better with our undomesticated glycine soja? And that's exactly what I looked at. So here we have ion leakage, and these are just different types of soybeans. So they're called cultivars, and we have from a 000 all the way to 7 of glycine max, and then a 0 to a 4 for glycine soja. And we're comparing the non-acclimated in the dark, those that were held at 22 degrees prior to this assay, versus the ones that were held for a week at 4 degrees C prior to this assay. And in every single case, we see acclimation, right? These clear bars are lower than the dark bars. And when we look at the northern versus southern, here's our northern variety, and then here's our southern variety. So there is a modest increase, right? When you are a 000, after these same conditions, you've got about 22% leakage, whereas Harbor's got almost 50. So there is a slight difference here. However, when we look at glycine soja, it's pretty stable. This dampened our enthusiasm for would there be cold-tolerant genes present in this undomesticated population. When what we're seeing is no matter where they're coming from glycine soja, they have about the same leakage, and it's all within the same range that glycine max has. So there's probably not good genes here. Okay, well now we got to go back to the drawing board. So we've thought back to, okay, what do we know about cold tolerance? What is the molecular control of this? And for that, we go to Arabidopsis. Again, Arabidopsis is a cold-tolerant plant, and this is how the pathway works in Arabidopsis. This pathway is called the CBF cold-responsive pathway. If this primary transcription factor, CBF, is knocked out, the plants cannot acclimate and cannot survive the cold. So it controls 15% of the genes, but they're 15 critical percent, because without it, it cannot survive. when you look at Arabidopsis, exposure to cold increases cytosolic calcium levels. This activates a kinase cascade, and then SEAS1 stimulates ICE1 to prevent its degradation by HOS1. OST phosphorylates ICE1 also to prevent its ubiquitination by HOS1. And then ICE1 is a transcription factor which activates our CBF-DREBs. From there, CBF-DREB go to activate CRT-DRE containing elements. So this is just the consensus sequence that it seeks out, and it's found in most of the cold-responsive genes in Arabidopsis. And then you get the process of cold acclimation. So we know all this is true in Arabidopsis, but what about soybean? Is the fact that soybean's cold intolerance potentially due to they don't have homologs to these genes? Are these genes just not present and or not working? And Dr. Yamasaki looked at this a couple years ago, starting with the dihydrins. So this is a family of cold-responsive genes that's very important. When you lose dihydrins in Arabidopsis, you lose cold tolerance. So it was a good place to look for homologs in soybean. And he found homologs in soybean, but they did not increase, the transcript levels did not increase in the cold. So these are not increasing in the cold. So at least from here down, this pathway is definitely not working, even though the genes are present. So he took a step up the pathway and did a search for homologs to the CBF-DREBs, so from Arabidopsis in soybean. And he found 7. GM stands for glycine max, and AT stands for Arabidopsis thaliana. So there are 4 Arabidopsis CBF-DREBs, and then there were 4 soybean CBF-DREBs that were the closest homology to Arabidopsis. It was those 4 that the lab kind of moved forward with to see, okay, they're there, are they activated appropriately? If they're cold responsive genes, we'd expect to see the transcripts increase just like you see in Arabidopsis. And that was the next step. In soybean, when you measure the transcripts of DREB1A and DREB1A2, and this is 2 different biological replications, and you're looking at leaves and stems, right? So 2 different organ tissues to see what's happening. The fold changes were massive in the early cold stress. So here, 1,000-fold, 400-fold, 450-fold, right? There was a large, massive increase in these transcripts. If you were to look at Arabidopsis transcript levels, this is exactly what it looks like. There's a really sharp increase in early cold stress, and then it comes down and maintains a slightly higher basal level while it's in the cold, but there's only a short burst. So this looks exactly like Arabidopsis. Okay, we're still not getting cold acclimation. We're still not getting increases in these CRT-DRE genes. So what's going on here? Well, next hypothesis, are these incapable of binding to CRT-DRE promoters? Perhaps over evolutionary time, they've just mutated where their active site no longer is able to bind to the right targets. So to answer that question, Dr. Yamasaki took 3 of these, and he put them in Arabidopsis under constitutive expression, so they were on all the time. And before we get too far into that, I want to show you what Arabidopsis looks like. So this is wild-type Arabidopsis, 0 hour, no cold. Wild-type Arabidopsis, 24-hour, after cold. And these are 3 genes that contain those CRT-DRE elements that CBF binds to. You can see all 3 are increased in Arabidopsis in the cold. And when you take those soybean CBF-DREBs, hook them to a constitutive expression promoter, and you look at these same transcripts without any cold exposure, those same transcripts increase in a similar fashion. So this suggests that, yeah, they are capable, at least in a functional cold system, the presence of these genes are enough to activate the downstream targets. And when you do freezing tolerance, they act like they're acclimated plants, even though they've never seen cold, right? You just take them and put them right in the freezer, and they respond just like an acclimated plant would. So right now, we know that soybeans are transcriptionally cold responsive in soybean. They go up really sharply and come down like they do in Arabidopsis. And when you put them in Arabidopsis, which is a functional cold tolerant system, they activate those downstream targets. So why is soybean cold intolerant? Could there be something outside of this pathway that's influencing it, that's making it maybe inhibited so that it can't respond quite exactly the way it should? Back to the literature, where I was able to locate IN3. So this is IN3 and its homolog EIL1. When they're frozen without any exposure to cold, you see a lot of death, right? They don't acclimate well. And when you look at transcripts, here's the CBF proteins, then here's those downstream targets. At 22, so this is at normal temperature, in these knockout plants, all of these transcripts are upregulated, suggesting that this does have a negative control. And this paper went on to show that, yes, IN3 binds to the to its motif in CBF. So at least in Arabidopsis, this is happening. But we don't know much about soybean. Before we dive into soybean, just want to refresh everybody about ethylene. Ethylene is a very small molecule, but it regulates lots of stuff: growth, ripening, senescence, abscission of the leaves. And you can look here, right? You go from green to banana bread thanks to ethylene. And we know that it's involved in the stress response. So we want to look at it a little closer. When you look at the ethylene signaling pathway, you have ethylene, its receptor, a kinase, a stabilizer, a ubiquitin, E3 ligase, and a couple of transcription factors. This pathway is really cool to see in motion. So instead of stepping you through all this, we're going to look at it in a little bit of a cartoon fashion. In the absence of ethylene, so notice there's no ethylene over here, the ethylene receptor is keeping CTR1 activated. So it's phosphorylating IN2. This is a kinase, and it's phosphorylating IN2. Inside the nucleus, IN3 is being marked for degradation by EBF1-2, so it's degraded by the 26S proteasome. When ethylene shows up, it binds to its receptor. CTR1 is deactivated. This allows self-proteolytic cleavage of the C-terminal from IN2, and it moves into the nucleus where it stabilizes IN3 and disrupts this ubiquitination process so that it's no longer broken down by the proteasome. Now IN3 can bind to its targets, and then there's a couple more ethylene-responsive transcription factors, and then the broad suite of genes can be turned on. So why are we looking at all this, right? I told you I'd talk about cold, and here I am talking about the ethylene pathway. Well, remember, IN3 has also been shown to negatively regulate our CBF-DREB cold pathway. So now let's look at it in soybean. These are the different homologs in soybean to those same genes I just showed you from the ethylene pathway. So we have CTR1, ETR1, IN2, IN3, EBF1. And when you look at the IN3 family, these are transcript folds, 1 versus 0, 24 versus 0, and 24 versus 1 in Arabidopsis out here. And notice there's very little color change. Now we're going to compare those same times in these soybean homologs. And they are upregulating based on the darkness of our scale, right? If we look at this heat map, they're actually increasing, where they're not doing anything in Arabidopsis. The positive regulator, IN2, it goes up a little and comes down, but it goes up a lot